Good morning, students. So let's continue with the general microbiology. Today's topic is morphology of bacteria. Morphology of bacteria. So actually the objectives, learning objectives of this particular class. So these are the objectives. Classification of bacteria based on shape, arrangement and gram staining property, as well as bacterial anatomy. Structure of bacteria including the cell wall, cell membrane and cytoplasmic matrix. And this topic, it comes under the competency MI 1.1. Okay. So let's see the morphology. Morphology of bacteria. First of all, size. Size of bacteria. So bacteria, as you know, this is microscopic. As you know, all the microorganisms are cannot be visualized with your naked eye, so you have to depend on microscope. So all these are microscopic or microorganisms. And next is a shape and arrangement and motility, as well as the detailed structure or bacterial anatomy. All these things I will be discussing today. So coming to the size, the unit of measurement used in microbiology or bacteriology, that is microns or micrometer. Microns or micrometer. And 1 micrometer, as you know, it is 1/1000th of a millimeter. 1/1000th of a millimeter. And we can take the case of viruses. Actually, it is even smaller than bacteria. They are ultra microscopic. You can't visualize this viruses using the ordinary light microscope. In that case, you have to depend on electron microscope. And the unit of measurement in virology, it is nanometer, which is 1/1000th of a micrometer. And the bacteria of medical importance, so they are of this size. The diameter, it varies from 0.2 to 1.5 microns and the length 3 to 5 microns. So this is the size, normal size of all the medically important bacteria. And again, as we have discussed in the introductory class, so you must be remembering about Leeuwenhoek. So Leeuwenhoek is the first person who observed bacteria under the microscope. And coming to the shape, so depending upon the shape, you can classify bacteria into cocci, bacilli, then comma-shaped ones. So spherical bacteria, they are otherwise known as cocci, and rod-shaped ones are bacilli or bacillus as a singular. Then comma-shaped ones are there, example Vibrio cholerae, which is a causative agent of cholera. Then spiral bacteria are also there. Rigid spirals as well as flexible spirals are there. Flexible spirals, examples spirochetes, under which this Treponema pallidum, which is the causative agent of syphilis, then Leptospira, causative agent of leptospirosis. So all these are the examples of spiral bacteria. Then filamentous and long branching bacteria are also there. So they are actinomyces. And bacteria without any particular shape are otherwise known as mycoplasma, because they don't have cell wall, so they can acquire any shape. So these are mycoplasma. So based on the shape, you have spherical ones, cocci, then rod-shaped ones, bacilli, then comma-shaped, spiral forms, etc. So these are cocci, then bacilli or rod-shaped bacteria, then spiral ones, spirochetes, namely Treponema, Leptospira, Spirillum, all these are the examples of spiral bacteria. Then comma-shaped one, Vibrio cholerae, then actinomyces, long filamentous, lengthy ones, these are actinomyces, and Mycobacterium, which is the causative agent of important diseases like tuberculosis, leprosy, etc. So they are also long and lengthy, just like fungus. That is how they got the name Mycobacterium, similar to fungus. The next point is arrangement. So after the shape, size, then now I am coming to the arrangement, bacterial arrangement. So again, you can have different types of bacterial arrangement. Sometimes bacteria can be seen in singles or in pairs or chains or also in clusters. So see, these are the bacteria seen in singles, then Diplococcus or in pairs, then Streptococcus or in chains, then tetrads. 4 bacteria are arranged together in a cluster, so that is tetrads. then cluster, bunch of bacteria, just like a bunch of grapes. So that is seen in Staphylococcus. Then Sarcina means it's a group of 8. So all these are the different forms of bacterial arrangement, different types of bacterial arrangements. Then another peculiar type of bacterial arrangement which you can see in different bacterium, that is Corynebacterium diphtheriae, which is the causative agent of diphtheria, that is Chinese letter pattern arrangement. Chinese letter pattern arrangement. So bacteria or bacilli are arranged at an angle to each other. So they are seen in V forms, L forms, etc. Okay, so these bacteria, since they are too small, you have to depend on the microscope, and under the microscope again it is very difficult to observe the bacteria. So we have to again depend on various staining techniques. Okay, so these staining techniques will enhance the visibility of bacteria. So there are so many different types of staining methods. So these are simple staining, differential staining, then negative staining, as well as certain special staining techniques are also there. So under differential staining you have two important staining techniques like gram staining as well as acid fast staining or Zn staining. So these are very commonly or routinely done in our laboratory. Then special staining techniques are silver impregnation staining, capsule staining, spore staining, etc. So about all these things you will be studying in detail in the coming classes. So gram staining, actually it's a very basic thing, and every lab, microbiology lab, there is a basic requirement, and this is a practical exercise also for you. This is a hands-on training will be there on this gram staining. So I will not be telling too much about this, but just give you an idea about what gram staining is. So here this is, as I said earlier, this is a differential staining. There are different dyes and different steps are involved in the procedure. So there is a primary stain, then a decolorizing step is there, then next you have to go for a secondary staining step also. So as primary stains we are using crystal violet or methyl violet. Then acetone is the decolorizer. Then followed by the decolorization, you have to stain the slide with a secondary stain, which is either safranin or carbofuran. Okay. And based on this gram staining, you can categorize or group bacteria into two major groups or two major types, namely gram positive and gram negative bacteria. So gram positive bacteria, they resist decolorization and they retain the color of the primary stain, and they, finally at the end of the staining technique they look purple or violet. Okay, whereas gram negative bacteria, they lose the primary stain during the decolorization step and they take up the color of the secondary stain, which is safranin, and they appear pink. So this is the observation. In the end of the staining, either the bacteria will look blue or violet, or else it is pink. So pink ones are gram negative and blue or purple ones are gram positive. Okay, so these are the gram positive and gram negative bacteria. So in the first picture you can see the purple colored cocci arranged in singles and chains as well as in pairs, and in the second picture you can see the pink colored rod shaped bacteria. So the second one is gram negative bacilli and the first one is gram positive coccus. Okay, so why are they looking different? So both gram positive as well as gram negatives are undergoing the same steps of staining, but in the end some are looking violet and the other one is pink. So what is the reason behind this? See, there are some basic differences in the structure, especially that of the cell wall of the gram positive and gram negative bacteria. Actually, cell wall is the important structure which contributes to gram reaction. So based on these differences, so they lose or retain the primary stain. So that is the important step or important principle of gram staining. So we can explain this gram reaction using various theories like cell wall theory, lipid content theory, then acid protoplasm theory, etc. In gram positive bacteria, the cell wall is too thick and that will help the bacteria to retain the primary stain. Whereas in the case of gram negative bacteria, cell wall contains more of lipid. So your decolorizing agent is a lipid solvent. So during the decolorization step, minute pores are created in the cell walls of gram-negative bacteria, and the primary stain will lose out of the cell. So that is how they appear pink, by taking the color of the secondary stain. Similarly, the protoplasm of the gram-positive bacteria, they are more acidic in nature compared to gram-negative. So this will also help to bind the primary stain, which is more basic in nature. So these are the theories by which you can explain the gram reaction. Okay. So based on this gram reaction as well as the morphology of bacteria, you can classify bacteria into different categories. So with examples, I will be explaining this. So first of all, first group is gram-positive cocci. So first of all, arranged in clusters. Gram-positive cocci arranged in cluster. Example is Staphylococcus. Then gram-positive cocci arranged in short chains. Example, beta-hemolytic Streptococcus. Then again gram-positive coccus in long chains, again Streptococcus, virulent Streptococcus. Then gram-positive cocci arranged in pairs, example Pneumococcus or Enterococcus. Then pairs or in short chains. So there are so many different examples for each of these different types of bacterial arrangements. Again, you don't worry about this. So in the subsequent classes, you will be studying each one in detail. Then about the gram-negative cocci. Gram-negative cocci arranged in pairs. You have important bacteria like meningococci, gonococci, etc. So meningococcus, gram-negative cocci, these are arranged in side by side with the adjacent sides flattened, whereas bean-like appearance. Whereas in the case of gonococcus, the adjacent sides are kidney shape, or they are concave, so giving a kidney shape appearance or reniform appearance. So that is how you differentiate meningococcus from gonococcus. These are two important pathogens causing meningitis and gonorrhea, which is a sexually transmitted disease in humans. So these are cocci, gram-positive cocci and gram-negative cocci. Then coming to the bacilli, again you have gram-positive bacilli. I have already told you about the Chinese letter pattern arrangement or cuneiform arrangement, which you can see in the case of Corynebacterium diphtheriae, which is a causative agent of diphtheria. Okay, then bamboo stick appearance you can see in the case of Bacillus anthracis. So bacilli, gram-positive bacilli arranged in chains, giving a bamboo stick appearance, that is typical of Bacillus anthracis, which is a causative agent of anthrax. Then branched and filamentous gram-positive bacilli, these are actinomyces. Then gram-negative bacilli. Gram-negative bacilli, actually they don't have much difference in the types of arrangement, but again there are difference in the shapes. So pleomorphic bacteria, gram-negative bacilli, which are pleomorphic in shape and size. So these are examples are pneumococcus, pseudomonas, then protease example, etc. are examples for pleomorphic bacilli, gram-negative bacilli. Then comma-shaped gram-negative bacilli, example Vibrio cholera. Then curved ones, again spirochetes. Okay, so these are the pictures of gram-positive coccus in cluster, short chains, long chains, as well as in pairs. Then gram-positive cocci, then gram-negative cocci, kidney-shaped as well as bean-shaped ones, meningococcus and gonococcus, then gram-positive bacillus in chains and cuneiform arrangement. Then coming to the gram-negative bacilli, arranged in scattered form, then comma-shaped one and spiral forms. So these are all the common bacteria of medical importance. Then coming to the structure, detailed structure of a bacterial cell, bacterial anatomy. So this is the structure of a typical bacterial cell. So you can have the outermost covering. Outermost covering is the bacterial cell envelope, which is comprised of cell wall and the plasma membrane or cytoplasmic membrane. And in case of some bacteria, you can have one more outer covering, that is a capsule. So not all bacteria are capsulated. Some bacteria, they are capsulated and some others are non-capsulated also. So cell wall is the outermost cover in such cases. And inside of the cytoplasmic membrane or plasma membrane, protoplasm is there, and protoplasm contains various organelles. But unlike the higher forms of life, as in the case of humans and other animals, so bacteria, they lack several organelles. Like all the membrane bound organelles are absent in the case of bacteria, like mitochondria, endoplasmic reticulum, etc. are absent in bacterial cell. And again, nucleus also, there is no separation from the cytoplasm. Nuclear material lies freely within the cytoplasm. There is no specific nuclear membrane also. Okay, so this is the structure of a typical bacterial cell. And among these, the important one is a cell wall. So for your exam point of view, bacterial cell wall is very, very important. You have to study in detail about the bacterial cell. Similarly, this structure is also important. You may get a question like draw and label of bacterial structure. Then in that case, this structure is very important. Then going to the bacterial cell wall. Okay, bacterial cell wall. This is a tough layer which is thick, and it is of 10 to 25 nanometer thickness, and it weighs about 20 to 25% of the dry weight of the bacterial cell. And what are the functions? What are the functions of bacterial cell wall? So it gives protection. It gives protection to the bacterial cell. So it gives protection from various external environmental conditions like osmotic lysis. Then it protects from the toxic substances in the surrounding. And this is a site of action of the various antibiotics also. And it confers rigidity and ductility to the bacterial cell. And it also has got so many virulence factors. The bacterial cell has got so many antigens, and these antigens are important virulence factors of the bacteria. And antibodies are also produced against the bacterial cell wall antigens in the host. And what are the differences between the cell walls of gram-positive and gram-negative bacteria? So this is also very important. This is an important exam question also. So let's see what are the differences. Coming to the peptidoglycan layer, it is thick in the case of gram-positive bacteria, whereas in gram-negative bacteria, it is thinner. And pentaglycine bridges are there in the case of gram-positive cell wall, whereas it is absent in gram-negative. The lipid content is seen more in the case of gram-negative, whereas it is nil or scanty in gram-positive cell. The lipopolysaccharide is also absent in the case of gram-positive cell wall. Then teichoic acid is present in the gram-positive cell wall. Even though gram-negative cell wall is thinner, it is more complex and complicated because it has got variety of amino acids and proteins. Okay, so these differences are very important. So coming to the detailed structure of the gram-positive cell wall, the major constituent of gram-positive cell wall, it is peptidoglycan. Peptidoglycan, it is nothing but, it's a repeated unit of N-acetyl-muramic acid and peptidoglycan layer. It is too thick in the case of gram-positive bacterial cell wall, and it is made up of N-acetyl-muramic acid. N-acetyl-muramic acid and N-acetylglucosamine. So repeating units of N-acetylmuramic acid and glucosamine, it forms that is a backbone of peptidoglycan, and each of these layers are cross-linked with tetrapeptide side chains and pentaglycine bridges. So this is the structure of peptidoglycan. So you can see repeating units of N-acetylglucosamine and N-acetylmuramic acid, which are these layers are cross-linked with the tetrapeptide side chains. Okay. So this is the structure of gram-positive cell wall. So here you can see the thick outermost part, which is seen in red. So that is the peptidoglycan layer. It has got so many different layers, and along with that through that peptidoglycan layer, some hair-like structures which are projecting outside. So these are lipoteichoic acid molecules and teichoic acid molecules. So these are also very important structures of the gram-positive cell wall which are absent in gram-negative cell wall. Teichoic acid and lipoteichoic acid. These have got important functions in the gram-positive cell. And inside of the cell wall you can see the plasma membrane or cytoplasmic membrane, which is trilaminar as in the case of higher forms of cells. Okay. Then coming to gram-negative cell wall. So as I said earlier, this is thinner compared to gram-positive cell wall. And again it is made up of peptidoglycan as well as several amino acids and proteins. So peptidoglycan is similar to that which you can see in the case of gram-positive bacteria. It is made up of N-acetyl-muramic acid and N-acetyl-glucosamine moieties and cross-linked with the tetrapeptide side chains. So apart from this, there are so many other structures also in the case of gram-negative cell wall. So these structures are very important. You may get a question like gram-positive as well as gram-negative cell wall, or else just bacterial cell walls. So in that case, you have to draw and label both gram-positive as well as gram-negative cell wall and describe about the structure in detail. So gram-negative cell wall, here again, so this red colored two-layered structure, that is a peptidoglycan. So you can see it is very thin compared to that of gram-positive cell wall, and outer to this this peptidoglycan layer you can see another important layer, that is outer membrane protein. So this outer membrane protein is very important in the case of gram-negative bacteria, and the structure of outer membrane protein, that is very complex and complicated. So you can see outermost part of this outer membrane. So this is the outer membrane layer. This one is the peptidoglycan layer, and this one is the cytoplasmic membrane or plasma membrane. So the space between the plasma membrane and the outer membrane, that is otherwise known as periplasmic space. Periplasmic space in which lies the peptidoglycan layer. And on the outer part of the outer membrane, you can have another layer. So that is lipopolysaccharide, which is very important. Lipopolysaccharide, again it has got 2-3 different components. So this yellow colored round or spherical structures, so these are lipid A, and just above that you can see the core polysaccharide layer, and the outermost hair-like structures are the O-specific side chains. So these three structures together, this lipid A, core polysaccharide and O-specific side chains together form the lipopolysaccharide, which is very important in the case of gram-negative bacteria. And this lipid A, it has got the action of endotoxin. So gram-negative bacterial infections, you get endotoxic shock. So this lipid A moiety is responsible for the endotoxemia or septicemia in the case of gram-negative bacteria. It has got multitudes of actions. So in the coming classes, along with each of the different bacteria, you will be studying in detail about endotoxins. Okay, so that is a part of the lipopolysaccharide of the outer membrane layer. And apart from that, you can have another protein known as outer membrane protein, which is also seen on the outer membrane layer. So this also has got vital role. This helps the exchange of molecules. This outer membrane protein helps the exchange of molecules. And this one is the plasma membrane. So that is about the cell wall of the gram-negative bacteria. Okay. So lipopolysaccharide, very important. It has got 3 components: lipid A, core polysaccharide, and O side chain, or it is otherwise known as somatic antigen or O antigen. Then periplasmic space also is there. Okay. So the next structure is the cytoplasmic membrane. So inside the cell wall you have the cytoplasmic membrane or plasma membrane, which is somewhat similar to that of the higher forms of life, the cells of other higher forms of life. but only exception is sterols are absent unlike the animal cells. Sterols are absent in bacterial cytoplasmic membrane. Otherwise, it is same trilaminar structure and it is semi-permeable also, semi-permeable membrane. Okay, and what are the functions of cell membrane? So, it is semi-permeable membrane, so there will be exchange of the ions, then it acts as a transport system and it is site for various metabolic processes also. Okay. So, with that, I am winding up today's session and will be continuing with the rest of the bacterial cell structures. So, just to summarize, so today you have learned about the bacterial size, then different bacterial shapes, namely cocci, spherical bacteria are otherwise known as cocci, then rod-shaped ones are bacilli, then comma-shaped ones are there, spirochetes, spiral bacteria are there. So, these are the different types of bacterial shapes. Then you can have the different types of arrangement also, either singly or scattered or in pairs or chains or in clusters also. Accordingly, you have different bacterial examples. Streptococci are seen in chains, staphylococci are seen in clusters, and pairs you can have gram-positive as well as gram-negative bacteria like pneumococcus, meningococcus, all these are seen in pairs. Then about the gram staining also I have talked briefly. So, since this is a practical exercise, you will be learning in detail in the practical classes. Then about the cell wall as well as cell membrane. So, you have to study these structures in detail, these things in detail, because this is the basis of bacteriology. So, study well. Okay. Thank you.